So good afternoon, everyone. Good afternoon, everyone, and welcome to 19 Washington Square North, which is the home and intellectual center of NYU Abu Dhabi here in New York City. My name is Michael Pruganan. I'm a professor of biology here in the New York campus, and I also run a laboratory at NYU Abu Dhabi, and I have the pleasure of serving as the academic director here at 19 Washington Square North. So over the course of the academic year, here at 19 Washington Square North, we organized a series of public lectures, workshops, conversations, and other events that allow the public and the NYU New York community to learn more about various things that are going on around the world. Many of these activities are organized by the NYU Abu Dhabi Institute, led by the Director of Academic and Research Programs here, Sharon Hakakian Bergman, over there, who with her staff organizes many of the activities here. We should give her a round of applause for organizing these events. Today, we're very, very pleased to see all of you on this public lecture in memory by one of our esteemed faculty colleagues and friend of mine, Professor Tom Carew. Tom is a renowned neuroscientist, having been on the faculties of Columbia, Yale, and the University of California at Irvine, the latter of where he was the Bren Professor and Chair of Neurobiology and Behavior. He then moved to NYU in 2011 to serve as the Dean of the Faculty of Arts and Science, and was also appointed a Silver Professor. I should note he's also been active in the early years of NYU Abu Dhabi, visiting the campus numerous times, helping in developing its initial faculty and also teaching J-term courses there. Tom has made important contributions to our understanding of long-term memory and associative learning, providing mechanistic insights into learning and memory. He has written a major textbook on behavioral neurobiology and served as the president of the Society for Neuroscience, one of the largest scientific organizations in the world. Tom is a fellow of the American Psychological Association and the American Psychological Society, an elected member of the Society for Experimental Psychology, an elected fellow of the American Association for the Advancement of Science, and an elected member of the American Academy of Arts and Science. Again, it's a pleasure to welcome you all here this afternoon to this event, and let us welcome Professor Tom Carew.
Thank you, Michael. Do I need the mic? Can you all hear me the mic okay, or should I? Because I tend to wander around, so I'll do my best to stay there. If you can't hear me, just get your hands up. So, well, thank you for that introduction. Michael is an old friend and by the way, near the neighbor. And I'm delighted to have a chance to join you particularly in this place, because it's got a special history in Abu Dhabi. And some of the speakers that have preceded me are daunting. Anthony Appiah is one of the greatest minds on the planet, and he gave a talk here last year. I came to, and so I feel really privileged to be part of that company. So what I'm going to talk to you about today is making memories in the brain. Oh, quick sidebar. If y'all want to ask a question, get your hand up, it's fine. At the end, we'll have a Q&A, that's perfect, but I'm happy to also discuss anything you want to talk about as we're going along, so don't worry about it. So let me go up where I can see what this is. So how many of us in the room are educators at one level or another? Right, so you've all seen these kids, and many wonder what's going on in their brain. What the heck? So I worry about the guy in the far end who's chair, but that's what's going on in their brains, and that's what we're going to talk about today. Because I want to know what's going on in the brain too. So let's go speak out, if we say under the hood, let's look at the forming of memory. Memories come to us through our brain, through our senses. It's the only way it happens. There's another kind of memory that Michael studied, it's a genetic memory. That's a totally different concept even. The memories, your experiential memories, get into your head in lots of ways. You use vision, you use smell, through your taste, through the outside world. So your visual memory, everybody in this room can see the arch right now in your mind. Unless you really just came in from the West for the first time. But if you're doing it on First Avenue, everybody has that visual. And I can now ask you to see the Christmas tree under the arch, and you can see that too. That's the memory going on in your head that was put there by your visual system. It's stark and easy to evoke. And that visual memory is probably memories that we usually get around the world with from. But then there's an auditory memory that we also have in our heads. If I go da 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 da, most of you hear Beethoven's too. Beethoven would roll over in his grave because I did it justice to him, but only 4 notes got that whole symphony in your head. Just a few notes. That's another kind of memory. And musical memory is different than visual, both in its zip code in your brain and the way it's produced. And then there's olfactory memory. And they're the most interesting because they're diffuse, they're kind of weird. How many of you have read Proust ever in your life? All right, so he has a very famous account of his memories of his he talked about a Madeline cookie being dipped into tea, and that odor brought back his whole childhood. So, and you've all had that experience. I as a kid, and when I was in high school and college, worked in a warehouse at night, and I can still, if I smell a box of, a lot of cardboard, that whole summer comes back to me. Not a discrete event, the whole summer comes back to me. So our odor memories are, whoops, my goodness, our odor memories are very different than the other two memories we talked about. But they all are memories stored in our head somehow. So the good news is we're really good at forming memories. The less good news is our memories are often wrong. And that doesn't mean you're lying, and we're going to get to that. An incomplete memory is not the fact you're making it up. You just are not getting it straight in your head. It can be a part of a trick. I'll show you a few, and it can be devastating if we're talking about eyewitness testimony. So we'll chat about that in a bit. So today I'm going to talk about two different things. Briefly, a primer on how our brain works, and then we're going to talk about, by the way, my block, can you see if I stand here? You okay? Good. And then after we talk about our brain works, we're going to go into usually the visual system and talk about you can't always believe what you see, and you can't always believe what you remember from that. And I'm also going to tell you the leitmotif is that's not a bad thing. We'll return to that in a little while. So first, let's talk about this. Here's your cortex. For most, if you're facing that, the front of the brain is to your left, the back of the brain is to your right. All those wrinkles are intentional. Nature's produced that. We'll talk about that in the middle in a minute. At the bottom, I can't point because the pointer works, so I'll just mention, at the bottom, that large structure is the cerebellum. That's what handles your balance and your stability. And then out of that cerebellum, there's that long kind of skinny thing coming out, number 22, that's your spinal cord. And that's where the brain talks to your limbs. And for example, watch this. Right? Can you see that? Right now I have action. And do you all know what an action potential is? It's kind of an electrical signal in the brain. They're leaving my brain at 120 meters per second. That's a football field and 2 end zones in 1 second, going down to the muscles of my fingers, releasing transmitter onto those muscles and making it. And that, by the way, is called, for the three aficionados, metochrony. This is metochrony. Just like swimmerets in a lobster. Metochrony means that there's one chronos, one time superposed on another, and my brain is sending those signals down that rapidly to make that happen. So now let's unpack this. There's functional regions in the cortex. These two are really important. The left to your left is called Broca's area, and the Wernicke's area is to the right. Those are areas involved in speech. Hugely important. Our ability to speak and comprehend speech is a primal kind of descriptor of our humanity. So knowing where those descriptors or where those areas are is very important. Now, it turns out that on the left-hand side, Broca's area, if you have a lesion there or a tumor there, something wrong there, what happens there is you can't produce speech. You can understand it fine. You just can't produce it. The flip side, over on Wernicke's area, you can understand, you can produce speech, but you just can't understand it. So what that tells you is there's different neural real estate that is assigned different functions in speech. And it's really important if a neurosurgeon, by the way, how many are right-handed in the room? Most of us? How many lefties are you? Okay, of the lefties, all of us right-handed guys, the speech areas on the left side of our cortex, like that. For the lefties, probably 70% of you are like us, but about 30% of you have your speech on the other side, in the right hand. The reason I'm saying that is not to be cute. It's that if you've got a tumor, and a neurosurgeon is going to go in and take that tumor out, she better know where your speech area is. Yeah. It's going to be critical. Has anybody ever heard of the WADA test? No. All right, it's really cool. Google it later and check it out because it's amazing. The WADA test is the way you tell which side of your brain has speech. So in your neck, you've got two huge blood vessels called carotids, left and right, goes up to different sides to your brain. And they're huge. They're the biggest blood vessels we own, all right? If you want to do something to the brain, you can inject something in one side, and that whatever that substance is will go into that side of the brain. So the WADA test is, I'm going to give it to you, is, let's say they're giving it to me. They would put a cannula in my left carotid. They would inject the equivalent, it's called sodium pentothal, something that transiently stops neurons from working, all right? And I'll tell you what happens. The neurosurgeon, she would say to me, all right, start counting, and then they inject. And I would go 1, 2, 3, 4, 14, 15, 16. Happens every time. It's amazing. That's because the brain was put to sleep transiently, and as it passed through my speech area, I no longer could produce speech. I was doing the right thing, but the brain didn't harvest it. Now she knows, okay, Tom's stuff's on the left side. If you're a lefty, you really want to know that if you know where the surgery is to be performed. So that's the WADA test. Turns out there's a very interesting thing for many people called bilingual aphasia. It is possible. Many of you are at least, Michael speaks Tagalog, I speak Spanish, and Alex speaks 15 languages. So the idea about this bilingual effect is you can actually lose one language and not the other. Typically, you keep your primary language. So I learned Spanish as a kid in Mexican playground in California. I could lose that and retain my English. So and that tells you that they're not identical pieces of real estate. Because if they all were in exactly the same circuit, then you lose it all. So you can lose one language and not another. So why do you think there's so many wrinkles? Anybody have a clue? Why do you think there's wrinkles?
Surface area. So
Perfect. When you pack to go somewhere, when Michael's on his way to Abu Dhabi, he folds up his socks and shoves them in his shoe. It increases the surface area. So if you unpack that, here's what you'd look like. That cortex would be the size of one of those giant pillows you get in a hotel. So it's a way to increase the packing area. You get more surface area by crowding it in, making those wrinkles. Good job, mate. All right. So it turns out that in the brain, this is a rough approximation. There's about 10 to the 13th neurons, a lot of zeros. Each of them, on average, has 10 to the 3rd connections. That's 10 to the 16th. That's a boatload of zeros. So think of the math. Think of everything you have to do to manage that number of synapses in the normal functioning of your life. Here's what a normal single neuron is drawn. It's in cartoon form. That T-ball in the middle is the nucleus. That's where the DNA lives. If you, I can't point. Maybe oh my goodness, I can point. Yeah, often with a laser pointer, the new kind of screens don't allow it. This is great. So this is a synapse. This is a single neuron coming down and synapsing right there. And if you look at that over here on the right-hand side, that synapse has a presynaptic element that's coming from the cell body down. Here, there's baggies filled with what are called neurotransmitters. Each baggie is called a vesicle. It's filled with neurotransmitter and it's packed in the presynaptic side of this. When an action potential comes down, that thing that comes 120 meters per second, they're really fast, comes down to the end. I won't go into the details, but calcium comes in and some of these baggies fuse to the membrane and open up. And they then dump their contents in their baggie out into the space. That, by the way, for 3 aficionados in the world, we call that an omega shape. It's like a Greek omega on its side. So when the baggie fuses, the contents, this is called exocytosis. And it's a highly conserved way. It happens in your brain, happens in a lot of other places in your body as well. So this is called a synaptic event when the transmitter is released from the presynaptic side, comes along, and it docks onto what's called a receptor on the postsynaptic side. And when that happens, these synapses are active. It turns out these synapses change with experience. So give me a minute. Who had a bike when they were a kid? Did you have a bike? What color was it? Red? Okay. My daughter's name is Amy. What's my daughter's name? Huh? Good, right? So you just have 2 memories in your head. One's about 15 years old and one's about 2 seconds old, and they feel the same. Amy didn't feel new. The bike doesn't feel old. But I'm telling you, the way that's made in your brain is totally different. So let's take a look. I got the color of your bike. I got my daughter's name now. We're going to talk about how her synapses just changed with that experience. And I'm going to talk about 3 things. We don't have to get into the heavy lifting on the nomenclature, but we're going to talk about gene expression, protein synthesis, and phosphorylation. The reason this is important is not because I want you to remember the terms. This is going on in your head right now. That Amy memory that we just put in her head, the bottom line just did that for her. So let's go into how it works. On the left-hand side is where the DNA is unfolded. Now, when you think of DNA, think of a rubber ladder, and then it's twisted on its long axis. So there's 2 sides, twist on the long axis, and each rung of the ladder is actually 2 pieces. AT is one, GC is the other. Anybody there in the room ever seen a kind of crappy science fiction movie called Gattaca?
yeah.
That's it. Gattaca are the nucleotides. ATGC, that's where it comes from. So what happens when you're going to what's called transcribe this gene, the DNA opens up, a molecular buzzsaw comes right down the middle of the ladder and separates it. So now the rungs of the ladder are sticking out here. And then along comes something called messenger RNA and matches rung for rung the code that the DNA had for it. I'm not, there's a little switch in there. I'm going to leave uracils talk to you, don't worry about it. So imagine each rung of this DNA ladder, half ladder, is now directly synthesized. And what's that called? Is transcription. When you transcribe a message, that messenger RNA is transcribing, literally, that's not a metaphor, it's transcribing the message from the DNA. Now, what are we going to do with that transcript? We're going to take it out to turn it into a protein. We're going to translate it. I'm going to go back over this and there's no quiz, so you're good to go. We're going to translate it. The way this works is very cool. There's these ferry boats called transfer RNA. They're not RNA that turns into protein. These are transfer, they're ferry boats, and each one of them has 3 little bumps. That's called it's the recognition tag for it to match onto the messenger RNA landing site. So this AAG likes to match UUC. So that ferry boat, this transfer RNA comes and docks on that, right? That's called a codon, that just matching AAG to the UUC. It comes in docks. It's carrying a cargo, 1 amino acid called phenylalanine. When it docks, it attaches that cargo to the other amino acid, the line aspartate, and you start to build. This is called the transcript, and that's called the protein. And we're building a protein. So the transfer RNA comes in docks. The cargo it has sticks it on to the proteins, to the amino acids that are there. Then it leaves, and the next one docks, and you build it. And now what I've described to you is if you take this long protein, imagine your grandma's pearls, right? A long strand. That's what a protein would look like? No. That's for the 3 aficionados in the world in here. That's a denatured protein. What a real protein would be like is if you took those pearls and lowered it into your hand. Now it has 3-dimensional structure. Now there's an inside and there's an outside. Some are close to each other, some are away from each other. And that's what a protein is like in your body right now and in your brain. And it turns out, and I'll tell you how do I change it? If I have that protein, watch my hand, and I stick phosphates on it, I can turn it into a channel. And I take the phosphates out and I stop it being a channel. I rephosphorylate it, boom, I turn it into a channel. So what that is a dynamic change in the shape of that molecule. And that's how your proteins are changed to fit into particular places in your body and the like. It's a protein folding is a big deal. Now, up here, that phosphorylation, that's where the Amy memory came from. That was like 3 seconds old in your brain at the time. The only way I could put that in your head is by phosphorylating your neurons. Your bite memory, though, that came from the other side of this. The genes made a protein, the proteins got stored. So I've just told you kind of the molecular architecture of short and long-term memory. They're very different. You don't know it, I don't know it, that's great. I don't want to think about it, but nature's given us this kind of amazing ability to make our proteins turn into different shapes. Now we're going to go into the kind of the brain itself, and this is just going to be a quick magical tour of looking at the neurons. There's more neurons at a cubic millimeter of cortex than everything you see right now. They're really packed tight. And so we're going to go through a quick tour through the universe, and this is just for fun. Has anybody ever heard of the rainbow mouse? All right. Well, it turns out that it was developed by a guy at Harvard. Actually, he's at Harvard now. He's at WashU, and his brother is in our biology department, Dan Sainz. So I'm going to do this just for the fun of it. We're going to go quickly, but these are ways to see neurons in color. And when you can see them in color, you can tell shape and projection and the like much easier than if you look, because if you looked at the brain, it looks like oatmeal. But when you color them, that's the cortex when you use these in this rainbow mouse. This shows you the beautiful array. This is the hippocampus. We'll talk about that in a while. Hugely important for memory. This is further in the cortex. These are cell bodies, and these are their projections. Can you see how beautifully arrayed? They're not just tossed like spaghetti in a bowl. They really have structure. And an important part of that structure is to know how these are all cell bodies. That's where the genes are. All on top are the input to those neurons. That's called dendrites. All of these projections on the bottom are all axons going out to talk to other cells. And that's just to give you a sense of how unbelievably complicated it can be. This is the brainstem, those are cell bodies and axons. Now we're getting into Lynn Keophas's area of vision, where we have pyramidal cells that are really beautiful. These columns are the way your cortex is built. The neurons are arranged in columns. That was discovered by Hubel and Wiesel and by Rosie Franklin, but they left her out. And what these columns are the structural computational units within the cortex These are cell bodies. That's where the DNA is. Up here are the dendrites and these beautiful flowers on top, and then they project out. And this is a single column. And there's a computational gain by putting things in columns. We can chat about that later if we want. On that single cell, every little yellow dot is a synaptic input. So you get a sense of how deep that synaptic input here. This is actually from a buddy of mine, Kelsey Martin. If you look at these, every dot, every green dot is a synapse. So you get the sense of, it's not just one cell talking to another, it's a boatload of input and a boatload of output. And yet the thing works. You can walk up and down stairs. It's pretty amazing. So let's talk about now how this machine we just talked about works. We've talked about, I'm going to develop two related themes, and I'm going to be in the visual system. We could use any. The visual system is the one that is most kind of easy to wrap your head around. So first of all, let's see if you can believe what you see. We're going to look through our eyes. The first thing is to understand how the visual system is arranged. This is the bottom of the brain, your two eyes. The retina is the light sensitive area inside each of your eye. Turns out that the outside, so since it's upside down, that the eye on the right-hand side is actually the left eye because it's upside down, but don't worry about it. On the right side of this eye, the retina is the light-catching sheet, and it sends information through this little intersection and stays on the right side of the brain. The other side of the retina projects too, but it crosses over and goes to the other side of the brain. And it's palindromic, your other eye does that. So what I'm really telling you is in your eyes, they both represent the same side of the brain, and they both represent the different side of the brain. And when you do an MRI, when someone's looking at it, you can see the cortex, this visual cortex in the back, really lights up by virtue of its activity. So now, let me tell you what you see. I'm going to tell you is not what you get. So can you read that? What is it? Anybody? Read that for me. It's an optical illusion, right? You cannot see optical if you're looking at illusion. You cannot see illusion if you're looking at optical. You flip back and forth. It's wonderful because you can see it, but you can't do it all at once. Your brain won't let you. There's a lot of stuff today you're going to find out that your brain won't let you do. So this one is an obvious one. What you probably see, as I do, are kind of a star and some Pac-Man. Turns out that there are no connected lines. Your brain's doing that for you. So if I took a light meter and went across this, the light would register there that much, that much, that much, and that much. It would see no line. Now I'm going to take a brain cell and I'll move it, and the brain cell then scans to here and it sees a line. The line's not there, but it's inferred from the whole scene, and it sees a line there, and then it stops seeing a line. Did it make a mistake? It's evolved to see lines, and we're tricking it to see a line. But if it didn't do that, you'd be tripping down the stairs as the way home. So that's how your visual system works. I'll show you some fun shapes and the like. Anybody ever seen the Necker cube? Really famous. It flops back and forth. It's one of the more famous kinds of things. I'm not going to take a lot of time to it. Look at the Ebbinghaus. So the ball in the middle and on the right here. Oops, my bad. So the ball here and the ball here. Which ball's bigger? Can you tell which ball's bigger? You cheated. They're the same. You went to this talk last year. Absolutely. I mean, it's clear they're the same. But what's amazing is the first time you look at it, they don't look the same. I mean, that guy looks tinier. This one's even better. Where do you see this one? Those two balls. The right-hand side to me looks smaller than the left. It's not. Here's one where I actually had to fight with a kid. I was giving a talk at Harvard, and the left table is longer than the right. And I go, well, and he said, nah, and he was going to get up and punch me out. So I took a line, I picked it up, and plopped it on the other side. And unless I do that, you know, not by now I'm playing with you, you know the left is the same as the right, but it sure doesn't look that way to your brain. And now, this one's fun, especially for people that are artists like Mary Jo in the room. So what color are those two lines? What color do you think the left line is? Take a shot.
Left line,
Yeah,
definitely darker.
and it's darker than the right one. We're positive of that. You got me money on you? All right, here we go. They're identical. They're identical. There's no darker lighter. They're identical. I take that away, I'm bump you, get it back. So you cannot, just because I tell you that, you can't change the way you see the world. And these are all parlor tricks. But later on, when we get into things like eyewitness testimony, it's no longer parlor tricks. So here's the thing. This is kind of my favorite. So let's see. So A and B, which is darker, A or B? Come on. Good. She says B, so too bad. She says B, so one says A, one says B, but we agree they're not the same, right? Do we still see that? I think you still feel that way. Now, wait a minute. Whoops. They're identical. They're identical. And I'm going to take it away and they're dark again. Because you can't lie. They're identical, but what are you doing? Why is that happening? It's happening because you're inferring a shadow from a cylinder. And you're inferring that that B can't be quite what it is because it's sitting in a shadow. But in fact, it's identical to A. So some of these are really cool. Now, this is going to be fun. On the sides, it doesn't work, but just bear with me. Stare into the, oh, we're not moving yet. These are, I'll get past these. Let's put it in motion. Just stare in the middle for a while. Are they moving for you? Wait till we get to the almonds. Someone's going to get seasick. Stare in the middle. Are they moving for you? Yeah. On the sides, it's not so good. Stare at the almonds. All right. And now you're ready for, have a swing that. Look at that and move your eyes around on the slide. Those wheels are rotating on you. They're not moving, but they're rotating for you. And what's happened? Oh, and this is cool. Focus on the X in the middle of this. You got it? Focus on the X. All right. So look at that black X and tell me what you see. Alex, what do you see?
Lighter.
Well, there's two Alexes. This is cool. Go for it. What do you see going around?
A pink.
Yeah. Let me see if you. No.
There's
Does
one-
anybody see a green ball going around?
Yes, at the beginning.
Most people, yeah, the green ball, and just keep staring, that green ball go around. There is no green ball. There was no motion. Neither was there motion or a green ball. Green ball came from the after image because he saw a red ball. And the motion was inferred by the sequential light. When you land on an airfield and you see the lights moving down, they're not moving. Sequentially, they're being lit and show you infer motion. So these are the way, by the way, it's not a parlor trick. This is the way the brain evolved. The important thing is to know that the motion was in your brain and that pink color was in your brain. It's hard for me to see it from here but often people see a green ball moving around by the after image. Now, this is good. Stare at that black dot. Give it 10, 15 seconds. Really stare at it. Just the black dot. What do you see? Wow. Yeah. Yeah. There's nothing on the slide, guy. Nothing on the slide at all. But it's there in your head. That's because that's an after image. You didn't see blue. You saw the complement of blue in your eyes, right? So if you stare that long enough, then boom, you'll see those four colors slides. Now, the next two slides are going to be easy. There's one slide and there's another. What did you see? What did you, I keep poking on you. What did you see?
I see a face. I mean, I see the individual parts as well, but I see a face.
So she's saying, perfectly said, she sees the individual elements like the bird, but you see the face. There's no face. You've learned what faces look like. So you imply that there's a face. We see three birds feeding little birds in the tree. But if you look at it another way, you definitely see a woman looking off to the left. Now, hold on. Read it out loud.
Only smart people can read this.
Actually understand.
Pretty cool, huh? You're not bad. It's amazing you can read that stuff, right? And the reason you can read is your brain is filling in the difference. There were no sentences, no words that made sense in that, but your previous experience really helped you get through that. So your previous experience is good for you, right? Always. I mean, who would say so? All right. As fast as you can, I'm going to show you just a bunch of words and I want you to tell me the color of the word. Not the word, the color of the word. Ready? Go. Out loud.
Red, yellow, green, blue, red, blue, yellow, green, blue, red.
These guys are good. All right, just one more time, just for the heck of it. Ready? Go. Duh.
Really?
The stoop gets you, that's called the stoop effect. I'm not going to go in a lot of detail, but we're used to getting meaning from words. We're not used to using the color as a real variable to pay attention to, but the meaning of the word. So it's very hard to only pay attention to the color. The bottom line is you're not in control of what you pay attention. That's a big deal. That's going to be important in a minute. Now, let's get into memory. Just a quick thing. There's three phases we talk about in the field. Get it in there, store it, and get it back out. One's called encoding, one's called storage, and one called retrieval. Let's talk about encoding. It turns out context really matters. This won't surprise you. Context really matters. So if I encode, let's say, the word jam in the context of traffic, and now I want to retrieve that word in the context of toast, it's very difficult. Same word, different meanings, different contexts. So as we form memories, we don't just stick them in there in a Rolodex, we're associating them with what we think we see. And different parts of your brain light up when you do that. The idea is that retrieval is an active process. Now let's get to the idea of storage. The retrieval error you made, it was not a retrieval error. It was probably because it was encoded differently. So when you're a teacher and a kid makes a mistake, maybe his or her mistake's not a mistake. Maybe what you're doing is you're asking them a question in a way that brings out the incorrect response. And because she or he is encoded it. Now, if this is pattern, anybody play chess? Who's the chess players in here? Okay. And if a chess master and a doofus looks at that, and a beginner looks at that, the chess master will immediately remember one side and not the other. Which one will he remember? Yeah, there's one side that's legal and the right side's illegal. And the bottom line is the patterns will matter to a chess master or to a non-chess player, even though the visual input's identical. Let's talk about this. Just bear with me. I just want to read something to you. Can't see, that's all right. Are you ready? Listen. We good? Thread, sewing, haystack, sharp, point, syringe, pin, pierce, injection, knitting. Got it? Okay, how many heard me say the word syringe? Thank you. How many heard the word me say sewing? How many heard me say the word needle? Keep them up. Keep them up. Keep them up. No needle. You know what happened. It's a parlor trick, right? You know what happened. When we get to eyewitness testimony, I saw him come out of the bank. Now it's not a parlor trick. But that is an associated mistake everybody makes. Why? Because I got so many other things going on that it sounds right. So I swear I heard it, and you didn't. And you didn't. Okay. Now let's get into the things of associative memory. What I'm really telling you, they're not facts in the Rolodex. They're reconstructions. If you take home anything, your memories are not, you pull it out and look at it, they're reconstructions of your past. And we're really good at it, but not perfect at it. So let's take a look at this. There's different kinds of memories we talk about. This is just in my trade. There's declarative and procedural memories. So declarative memories are what a bike is. If you can, in your mind, you know, if I say bike, you know what a bike is. That's a declarative memory. If I ask you when's the last time you rode a bike, you can remember that. Right? But that's still considered a declarative memory. Then there's a whole other kind of memory called a procedural memory. That's how to ride the bike. Very different memory. I'll tell you a story, a true story. How many of you drive a stick shift? All right. I grew up as a kid in California, only driving a stick shift. All I ever did. So the first time I went to an airport as a young adult, got in a rental car and turned it on, it was an automatic. And I started pawing for the phantom clutch. That's not there. I did not paw at the phantom clutch when I got in the car, or even when I sat down. It's when I turned on the car and it was context specific and there should have been a clutch there and there wasn't. Those kind of motor memories are routine. They're built, you can see that you can even think of your own kind of memories it is. But the bottom line is that memory is very dependent on the context you're in. And sometimes you'll have, and I'm going to show you a case of amnesia in a minute where that is lacking. So as a matter of fact, we'll get to it now. Do you remember that I drew the brain for you? This is called the temporal lobe. It's on both sides. There's a very important structure in the temporal lobe called the hippocampus. It's named that because it's shaped kind of like a seahorse. And it turns out that that's terribly important for memory. And we can know that by doing research, but we also can look at the tragedy of clinical studies where that's impaired. So I'm going to tell you now, how many have ever heard of HM? HM is a very famous patient, probably the most famous patient in memory research. We only could call him Henry Moilson after he passed in 2008. So I've been teaching this stuff for years. When I was at Yale, we didn't know who Henry was. We called him HM. It's just respect when you have a clinical patient. But HM, Henry Moilson, he was born early about 1926, and he died in 2008. And here's what happened to HM. And this is all true. This is not trying to be clever. Henry was, when he was a kid, fell off his bike and really whacked his head. And he had intractable seizures every day. I don't know about seizures. I've got a buddy of mine at NYU that studies seizures in kids. Some of them have 50 a day. So we're not talking about, oops, a bad day. We're talking about a life change. So Henry had intractable seizures. Now remember, this is early on. It's 1953. He was referred to a very famous neurobiologist or a neuroscientist at Hartford named William Scoville. And what Scoville did was remove his hippocampus bilaterally because that's where the seizures were. All right. Now, nobody wanted to hurt Henry, but they wanted to save his life because he couldn't get around the world. So these are absolutely true clinical cases that after the fact teach us a lot. So what happened to HM is after they cured the seizures, he had no more seizures, but he had horrible amnesia. There's a very famous, wonderful woman, Brenda Miller, who was at Harvard at the time. She would drive to Hartford 3 times a week to see him for years. She'd walk in, hi, Henry, how you doing? I'm doing great. Do you know my name? Nope. Don't know my name. Come in the next day, you know my name? Nope. Don't know my name. He'd never know her name. And yet Henry could learn other tasks. There's one called, anybody ever seen the mirror task? So there's a star, and all of us can trace a star easily with a pencil. Try it looking in a mirror. And it's weird because it's backwards. You all can learn to do it. You get pretty good at it pretty quick. Henry was fine at that. So what does this tell you? There are different memory systems in the brain. It ain't one computer. There's completely different memory systems in the brain. So the observation was Henry lost all his declarative memory, but his procedural memories were perfectly intact. Now that's not the way we want to find out about memory. But if you can learn from the tragedies of different things that happen in the world and take advantage of it, you can build models that help the next generation study. So the bottom line here is that there are multiple memory systems in the brain. There's recent memories stored in the hippocampus, and there's remote memories in the cortical circuits. And now we're going to get to false memories. I've already given you one, the needle trick. Somebody put your hand in the air. Hey, there ain't no needle on that. So you had a false memory. I put that in your head. Flashbulb memories. Back in the day, I could say, where were we? A lot of us could say, where were you when Kennedy was shot? And then tragically, we had to say which one, because 2 have been shot. But a lot of us can say, yeah, I knew where I was on that mine. That's a very common thing in memory is where were you? I'm going to tell you about Dick Neiser's study on the Challenger disaster. But another woman was here at NYU. Liz Phelps did this for the Challenger for 9/11. So let me tell you about Dick. Dick Neiser study. Does anybody know about the Challenger disaster? Part of the room does? Okay. When I used to talk about this, I remember Mary Jo may have been with me. I gave a talk once, and I said, just watch my hands. What does that bring up? And someone thought I was one of the village people. Right? Wrong image. But this contrail separation was visually very striking. And what happened was, less than 2 minutes into the flight, the whole thing blew up on the pad. And how many were there? 7 crew members, some astronauts, a payload specialist, school teacher. And that was a bloody disaster that was horrible. Dick Neiser was a psychologist at Emory University, and he took advantage of that. He took advantage of that horrible thing happened. And the next day, went around to every kid he could find on campus. Just if I went out at NYU and went all over the park, to every kid, what were you doing? Let's write it down. What were you doing? What were you doing that day? Let's write it down. So on that day, he took the biography of everybody he could find. I'm going to make up 100 kids, right? They're all college kids. And he got them the day after it happened. Then he went and chased them down 5 years later. I'm making up the story. It could have been 6 years. But anyway, he chased them out a lot longer. Now, I'm going to tell you the story as if it's me and Mary Jo, my wife, to give you the idea, but this is what happened today. So Dick finds me. I was at Emory at the time as an undergrad. Now I went to California to go to graduate school, and he chased me down 5 years later. They said, Tom, what were you doing on the day the Challenger happened? And I would say to him, well, I was leaving chemistry lab. I met Mary Jo, my girlfriend, at the fountain. We always sat at the fountain. We had lunch together. She opened up a sandwich. We had tuna fish sandwiches. There was celery. I don't like celery in my tuna fish sandwiches. The reason I'm saying that is my recounting is really granular. And then we walked together and we looked in the quad. We saw kids looking at the TV and this horrible thing on the TV, and it was devastating. That's my memory. One problem. I didn't meet Mary Jo until the next year. I'm not lying. I just didn't meet her until the next year. But we've been together 60 years. It makes sense when you put it together. What I just did was reconstruct that memory. No one's lying. You're making the best of it. But that memory I gave him was impossible. Now, it turns out I did meet her the next year, and I still don't like celery in my tuna fish sandwiches. So the memories are correct, but they're false. Now, it's a parlor trick sometimes to say it, but when we get eyewitness testimony, it's not a parlor trick anymore. So there's one, that's the nicer story. Let me just tell you one more story and then we're at the end. And this has to do with eyewitness testimony. Has anybody ever heard of Beth Loftus? Pretty well, okay. So Beth is really famous for implanting memories and studying false memories. And one of her famous studies, and I'm not sure if it's going to work on this, if it isn't, it doesn't, is to have everybody in this room watch the same visual scene, then put you in 4 rooms and ask you all what you saw with one word different in the question. So here's the scene. Good. Here's what everybody in this room sees. Okay, pretty dramatic. And now she takes somebody out and she says, I'm going to leave that there. And she says, how fast were they going when they collided, contacted, smashed, hit? The exact same words except one word, collided, contacted. And the estimate of the speed was phenomenally different. When she used the word contacted, it was 32 miles an hour. When she used just the word smashed, it was 41 miles an hour. So what I'm saying is pretty obvious is the way you ask the question is going to influence the kind of memory you get. And that has huge implications for things like implanted memories and eyewitness testimony. Where this really comes to be, any mom and dad in this room, is when little kids are being interrogated if something happened at daycare. And I'm not trying to be cute, but sometimes you really worry about, I'm not talking about getting beat up on the playground. I'm talking about abusing a kid. And they can give you a memory that puts someone in jail. And if it's true, throw the key away. If it ain't, you got a problem. So how you, a buddy of mine, Steve Cece, you've never heard, anybody ever heard of Steve Cece? No, he's up at Ithaca, New York, beautiful guy, and he studied memory. And he asked, one thing he did was he asked kids a question, young kids. And his own daughter was in this, have you ever been to the zoo? Nope. And he asked just that question every day for days. And he go, ever been to the zoo? Nope. You ever been to the zoo? Nope. By the 5th, 6th, maybe by the 7th, yeah, I've been to the zoo. By the 9th, yeah, I was at the monkeys. I loved it. And then we went and got ice cream. They're not lying. They're making stuff up because it's kind of fun. And if they're on eyewitness, they'll pass it. You know, that's a real memory. It just never happened. So why I'm being kind of glib but serious is when you think you remember something, you think it, but it may not be true. So the take-home message is memories can be absolutely real, but not true. So the last thing I want to tell you about is called reconsolidation. This has deep implications for therapy. So when you have a memory, you can read it on. When you have a memory, for some period of time after that, that memory is labile. You can disrupt it. We do it in the laboratory all the time. So you create a memory and then you distract somebody or you give someone a drug, different thing, and you can mess up the memory. In another group, you create a memory and you do that an hour later, nothing happens. That's called the consolidation period. What it means is it's the time during which that memory is labile, okay? And then that stops and now it's quote consolidated, the memory is secure. And so that was important to see. But what was interesting, and Beth was one of those that did this, is that reconsolidation happens too. So what that means is when you again bring a memory to mind, it's labile. Now, why might that be important? It could be a parlor trick like I've talked about, or you can have some Vietnam vet coming home having seen his buddy's leg shot off, and it's a memory just as soon not carry around with him. And if you can bring that back to mind in another kind of setting where it's a little more labile, what are we talking about? Therapy. We're talking about bringing that memory to mind in a safer context, not destroy the memory, but at least marginalize the memory in the context of one's life. So memories are labile, and we could take advantage of that in different contexts. So what I've just told you is memory is not as reliable as you think, is not as permanent as you think, and it's really a good thing. That's the way you want it. So now I want you to remember what I've told you. Oh, wait a minute. I've got one thing else to tell you. A brief coda. I'm asked a lot when I go out. We live in Maine and we go out to dinner with our buddies, and they'll say, and we all talk about what we do. There's lawyers and the doctors, and I'm a geek, and they say, well, can we improve memory? And it's a real question, and it's a real concern. Can we improve memory? So the question is, can we improve memory? And you at late at night see that ad? And the answer is absolutely no. No. I have hyphens with 30 words in that. No, because these prey on elders that are scared because they can't remember the kid's name. So they look for a pill. And you can tell I'm not neutral on this. I could probably get sued for this. I don't care. The fact is there's zero, you've seen the ads. This is going to make, and there's some pharmacists say, gee, I finally remember what my daughter's name is. No, It doesn't work that way. Prevagen might be nice to think about. It has no effect on memory. And there's a boatload of other drugs, I just picked on this one, that claim to improve memory. No, it doesn't. So what can improve memory? What can we do? First thing you do is get outside, go for walks, engage, keep your life engaged. Second thing you do is eat right. You can have a decent diet. Third thing is keep engaged in intellectual pursuits, play cards with your buddies, play chin or the like. And because Joe Quinlan's in the room, and I'm also Irish occasionally, you might have a pint of the black stuff. But the point is stay engaged. So if I'm going to end on anything, it's to keep your memory as sharp as possible. Just live your life. Thanks.